Thank you for letting me know. All right, hey, good morning and welcome to Biology Live Classroom. Today's topic is going to be BioPrinciples, and we're going to review the BioPrinciples diagram. The BioPrinciples diagram is in front of us right now, and if I zoom out, we can see the whole thing, kind of get a look at where we're going to go. We're going to start up here in the corner with cell theory. And then we're going to go to the characteristics shared by all living things. We're going to compare and contrast prokaryotes and eukaryotes, prokaryotic cells and eukaryotic cells. We're going to talk briefly about endosymbiotic theory. We're going to go through a list of organelles and their functions and talk about levels of organization shared by all living things, or the levels of organization of biology. So let's begin up here with cell theory in the upper right corner. Cell theory. Cell theory has three major points to it, and they are right here. Point number one, all living things are made of cells. If you're alive, you're made of cells. If you are not made of cells, you are not alive. Number two, the cell is the basic unit of structure and function. So if there is something doing work in nature, there's a cell involved. If there's something that has a level of structure to it and doing work, it's because there's a cell involved. The cell is the smallest unit that can do all of these things. That's what the word basic means, is basically, or basic means smallest. The cell is the smallest unit that can be alive and do things that living things do. That's what number two really means, is it's the smallest. Oops, thought I hit the button. There we go. Smallest thing that can do what living things do. Let's see if I can move this just a little bit here. So that's number two. The cell is the smallest thing that can do all the things that living things can do. Number three, the cell comes from pre-existing cells. Doesn't matter like what type of cell you are, whether you're kind of a simple prokaryote or a really complex multicellular eukaryote, all of your cells came from pre-existing cells, which is really interesting. What that means is you sitting there right now, you might be 2 or 3 or 4 trillion cells sitting there right now. If you were to follow these cells backwards in time, every one of those several trillion cells came from a pre-existing cell, and you go back a generation and back another generation. You keep going backwards until a time about 14 years ago when you were a single-celled organism. Everything that is you today was contained in a single cell. All the directions and instructions that made you what you are right now were sitting in a single cell too small for the eye to see. In fact, you could keep doing this. The entire human race can be traced back to a single human. Every organism on this planet, living thing that either was and is now extinct or is existing today, can go backwards in time until it was a single cell. In fact, all of this goes backwards until the only living thing on this planet was one single cell. So where'd that cell come from? That's a good question. Let's move this over a little. Wow, this is not cooperating with me. All right, that gets us where we need to be. So we go from cell theory then to the characteristics shared by all living things. Well, we remember this by using chara. The word characteristics, if you take the first 5 letters of characteristics, you have chara, C-H-A-R-A. C for cells, H for homeostasis, A for ATP, R for reproduce, in the final A, we put an N in front of it, nucleic acids. So let's do these all in order now. Cells. Well, we already learned from cell theory that all living things are made of cells. So if you are living, it doesn't matter if you're the simplest prokaryotic bacteria or some archaea cell, or a multicellular eukaryotic organism, you're made of cells. If you're living, you're made of cells. Number 2, all living things have to be able to respond to change. And that's right here in the word homeostasis. And I kind of missed on my arrow. Try this again. My goodness. So let's see if I can get us back to where we're supposed to be. And we have homeostasis and evolution. Respond to change. What is homeostasis? It's maintaining internal conditions. Well, if you're maintaining internal conditions, that means there's something out there trying to change them. So a living thing has to be able to adjust, respond to changes in the environment, or it won't be a living thing anymore. And things that aren't living can't change in response to the environment. So if you're responding to change in the here and now, that's homeostasis. If you're responding to changes over long periods of time, that's evolution. So we use H in chara to help us remember the word homeostasis, and homeostasis is supposed to help us remember, my goodness, this thing is all over the place with me today. I'm losing this battle with it. Okay, homeostasis is supposed to help us remember respond to change. Respond to change. Homeostasis is supposed to help us to remember respond to change. I think this thing is getting squirrelier as time goes on, so I'm not sure why, but now I can't even touch the screen without it going all over the place. Now let's see if we can continue. CHA, ATP. So in chara, the A is supposed to remind us of ATP. ATP is cellular energy. Well, what specifically does that mean? Well, let's compare it to your car. If you want your car to do work, you need to put gasoline in it. If your car doesn't have any gasoline in it and you try turning it on, it's not going to do anything. If you want your car to do work, it has to have gas. If you want a cell to do work, it has to have ATP. So gas is to a car as ATP is to the cell. ATP is cellular energy. So in chara, the first A is to help you remember ATP. It's cellular energy, and that's supposed to help you remember energy and materials. All living things need energy and materials. So let's start at the top. Cells. All living things are made of cells. H, homeostasis. All living things respond to change. A, ATP, energy and materials. All living things need energy and materials. CHAR, reproduce. All living things have to reproduce. If they're not making the next generation of living things, then whatever that living thing is will become extinct and it is no longer a living thing. CHARA, acids. The last A is for acids. What kind of acids? Nucleic acids. Name the nucleic acids. There are only two, DNA and RNA. All living things store their information on life, how to build and how to live in their DNA and RNA. All living things have DNA and RNA. If you do not have DNA and RNA, you're not alive because you don't have the information system to build proteins and do the things that living things need to do. So, CHARA, C-H-A-R-A, all living things do these. If, and this isn't a best 4 out of 5 kind of thing, you are doing all 5 of these or you are not alive. Let's move on. From the characteristics shared by all living things, we go down to prokaryotes versus eukaryotes. There are 2 types of cells in this world. There are prokaryotic cells and, wow, I don't know what just happened. There are prokaryotic cells and eukaryotic cells. So here's prokaryote and eukaryote. Now, this world is absolutely full of living things. You can dig down into the earth for a mile and still find living cells. You can go a mile up into the atmosphere and still find living cells, and every space in between them is full of living things. Though every living thing on this planet can be broken down into 1 of 2 types of cells. This planet is just an unfathomable number of living things living here, yet they are all either prokaryotic cells or eukaryotic cells. Well, what are the similarities and differences? If we're going to compare and contrast prokaryotes and eukaryotes, what do they have in common and what do they have that's different? So let's take a look. Now, prokaryotes and eukaryotes, if you were going to make a comparison, compare and contrast what's similar, what's different, you would often use or can use what's called a Venn diagram, and that's what you see in front of you. And notice with this Venn diagram, things that line up in both circles are things they have in common, and things that are outside of the overlapping area are things that are differences, or their differences. And I have 4 circles sitting outside of the Venn diagram. The first circle says size, and over here is multicellular, and over here is age, and over here is complexity. If you were to take the first letter of all of these, you would find SMAC, S-M-A-C. Well, when you have a sporting event and you have 2 different teams competing with each other, they have fans, and those fans want their team to win, and they usually talk smack about the other team and the other fans. And that's what you see here. You see 2 teams. You see the prokaryote team and the eukaryote team, and they're talking smack, and smack are their differences. Notice smack is outside of the center area. And since size is on the prokaryotic side, we're going to talk about size in terms of prokaryotes. Prokaryotes are smaller than eukaryotes. And age is on the prokaryotic side, and prokaryotes are older than eukaryotes. So when we're looking at this, prokaryotes are smaller by about 50 times. They're 50 times smaller. And prokaryotes are older. They're about 1 billion years older. There we go. So prokaryotes are 50 times smaller and about 1 billion years older than eukaryotes. And those are definitely differences. On the eukaryote side, we have these two words. We have multicellular and complexity. Well, you see, since it's on the eukaryote side, only eukaryotes can be multicellular. And that means they have more than one cell, many cells. Prokaryotes are only single-celled organisms. And eukaryotes are far more complex than prokaryotes because eukaryotes have organelles. And what are organelles but membrane-bound structures with highly specific function. So what are organelles? They're membrane-bound structures with a highly specific function. Eukaryotes have organelles, prokaryotes do not. And this is such an important point. Below the word complex is a list of organelles. Everything in this list is only found in eukaryotic cells. Prokaryotes do not have them. And this is tested repeatedly on the keystone, and kids still miss it. Everything on this list, underneath the word complex, underneath the word complex here, everything on this list, these are organelles, and they are only found in eukaryotic cells. So let's take a look at these organelles. I've broken them down into two types, plants and all other eukaryotes, or all eukaryotes. I'm sorry, I said that incorrectly, and I need to correct it right now. Plants and all eukaryotes, including plants. So I'm going to type that here because that's a really important point. Ooh, yeah, how'd that happen? Let's see if I can fix that. Of course not. So we have the arrow, and I should be able to do that, and it's being squirrely again. Let's see if I click this, if it's going to let me. Nope. This is supposed to say including plants. But I can't get back to it to finish typing the word plants. So you see the letter P there. All eukaryotes includes plants. So the first two that I'm going to give you up here are specific to plants. They're plants only. These are organelles found in only plants, and that's the chloroplast. Well, the chloroplast is an energy organelle that converts light energy to chemical energy in the form of glucose. So it takes energy in the form of light and it puts it into chemical bonds in a molecule called glucose, and it stores it in there like you would store energy in a battery. That's the chloroplast, and that's found only in plants. Plants also have a large central vacuole. Now this large central vacuole is just a storage facility like a refrigerator, and it holds water, and it holds anything that'll dissolve in water, such as amino acids and sugars and some nucleic acids, are all dissolved and stored in this large central vacuole. This large central vacuole, because it's large and it contains a lot of water, also gives a plant cell its turgor. That's a fancy word for saying it makes it tight. It makes the plant cell blow up right to its maximum size, and it can hold a lot of pressure because of the large central vacuole. Okay, so these two organelles, the chloroplast and large central vacuole, are specific to plants. Plants are eukaryotic cells, and as a subset of eukaryotic cells, they have these two organelles that other eukaryotes do not have. But all eukaryotes, so here we're going to do this again. All eukaryotes have everything in this list. So plants included, all eukaryotes, including plants, do Megan, and there should be a dash L here, Megan L. All eukaryotes, plants included, have Megan L, M in Megan for mitochondria. What do the mitochondria do? The mitochondria takes the energy stored in glucose. Remember, the chloroplast made glucose by converting or transforming light energy into chemical energy. Well, that chemical energy in glucose is then harvested and turned into chemical energy stored in ATP. Well, we've talked about ATP. Remember, ATP is cellular energy. Cellular energy is ATP. If you want the cell to do work, it better have some ATP. The mitochondria takes the chemical energy stored in glucose and transforms it to chemical energy stored in ATP. Next, ME. The E's for endoplasmic reticulum. The endoplasmic reticulum has two flavors, rough and smooth. The rough ER is rough because it's studded with ribosomes. Now notice ribosomes are not on this list. Ribosomes are on the very mad list. Ribosomes are shared between prokaryotes and eukaryotes, but the endoplasmic reticulum is not. The endoplasmic reticulum is an organelle and only found in eukaryotic cells. The rough endoplasmic reticulum is rough because it has ribosomes. And ribosomes are found in both prokaryotes and eukaryotes. What do? Ribosomes make protein. Well, the endoplasmic reticulum has a second flavor called smooth ER. And the smooth ER is smooth looking because it doesn't have ribosomes. Instead, it looks like tubes, kind of tubular. And the smooth ER makes lipids, fats, oils, and waxes. So this endoplasmic reticulum is rather important because we see a lot of protein synthesis in one protein in particular, enzymes, hugely important. And it sees lipid production, also absolutely critical. 2 of the 4 major macromolecules of life. All living things are made of these 4 macromolecules. 2 of those 4 macromolecules are synthesized right here at the endoplasmic reticulum. So Megan, or Megan dash, darn it, L. Remember, it should be Megan dash L. Oh, did it again. Megan dash L. So Megan L. We talked about the mitochondria, the endoplasmic reticulum. So it's ME. Here's the GA. Golgi apparatus. The Golgi apparatus is an organelle, a membrane-bound structure with a highly specific function. The GA, the Golgi apparatus, is an organelle that modifies and packs proteins. When the endoplasmic reticulum makes a protein, it actually only makes a poly something chain. And the word escapes me for some reason right now. Polypeptide. That's the word I'm trying to come up with. Let's write that out. We'll put it right there. Polypeptide chain. So you see, the endoplasmic reticulum, the rough endoplasmic reticulum, when it makes a protein, it makes a polypeptide chain. Well, that chain has to be folded. In fact, it has to be folded very specifically in 3 different ways before the polypeptide chain is an actual functioning protein. While the endoplasmic reticulum makes the polypeptide chain, it's the Golgi apparatus that actually folds it and modifies it and turns it into a functioning protein, and then it packages it up functioning protein and ships it somewhere, wherever it needs to go. So we start up here with the endoplasmic reticulum and chef ribosome. And chef ribosome, being a chef, reads recipes. What recipe? Messenger RNA. Messenger RNA is the recipe sent to chef ribosome. Where do you find chef ribosome? In the rough endoplasmic reticulum. Yes, it's a kind of a rough neighborhood, but it's where the best protein comes from. So, chef ribosome makes the polypeptide chain, and he sends it to his very good friend, mailman Golgi, who uses his apparatus to modify and pack that protein, that polypeptide chain, modifies it and packages it into being a real protein and ships it down the cytoskeleton subway to wherever it needs to go, mailman Golgi. And finally, that gets us to N, oh, I did it again, in Meganell. And the N is for nucleus. Now, you may have been told that the nucleus is the command center of the cell. It is like the mayor of the city, who's calling all the shots, telling his minions what to do and how to do them. I don't like that definition because that really doesn't capture what the nucleus does. The nucleus is more like a library, and this library contains chromosome books. Well, libraries contain books, and this library contains books. They're chromosome books, and these chromosome books, like other books, have chapters. They are gene chapters. So each chromosome has multiple genes, just like each book has multiple chapters. And these gene chapters contain the recipe. Each chapter contains one recipe for a single protein. Sometimes these proteins are enzymes, and sometimes these proteins are for structural purposes like muscles or collagen or cartilage. These proteins take on a lot of different functions, but how to build the protein comes from the nucleus, comes from a set of books in the nucleus called chromosomes, and these books are reference only. They can't be checked out of this library called the nucleus. The books stay there. But we do make copies of the recipes, and we send those copies out to chefs like Chef Ribosome. And these copies are called messenger RNA. More on that later. So in Meganell, the final letter is L for lysosome. And the lysosome is near and dear to me because it's kind of like the stomach of the cell. And I like to eat, and well, so does the cell. And the lysosome contains digestive enzymes. And when the cell eats something, those digestive enzymes, this lysosome, kind of engulfs it and bathes it in the digestive enzymes, breaking it down so the cell can use it. These are the organelles of the cell. And one more time, these organelles are eukaryotes only. They are not found in prokaryotes. But what are found in prokaryotes and eukaryotes? Well, we come back here to pro versus you, this competition we have going, where the fans are talking smack, size, multicellular, age, and complexity, smack, about the other team and their fans. Well, when people talk smack to each other, invariably somebody gets very mad. If you look carefully at the similarities, both sides are going to get very mad, right? Their fans are going to get very mad because the other side's talking smack. And how do we spell very mad? It's V-E. It's R-I. Yes, I know that's not very, but we're going to call it very. V-E-R-I-M-A-D. Very mad. Very mad C-C to finish it off. So, very mad C-C is what we have in common. Prokaryotes, did it again. Prokaryotes and eukaryotes, what do they have in common with each other? Very mad CC. The VE for very mad, vesicles. These are just simply storage structures, membrane-bound Ziploc baggies. A vesicle is a Ziploc baggie inside of the cell. RI, ribosomes. We already talked about ribosomes. If you were a eukaryotic cell, you would find these ribosomes embedded in the rough endoplasmic reticulum. But if you're a prokaryote, the ribosomes are just kind of floating around in the cytoplasm. Free-floating ribosomes. What do ribosomes do? They make proteins. So you have vesicles, Ziploc baggies, ribosomes that make proteins. Both prokaryotes and eukaryotes have those. We have cell membrane. The cell membrane, every living thing, every living cell has a cell membrane. We have ATP. All living things use ATP and DNA, both of which are part of Cara up above. And the CC, cytoplasm and cell wall. All cells have a cytoplasm. It's the jelly-like material inside the cell membrane that holds the other structures. And a cell wall is specific to just certain types of cells. So both eukaryotic cells and prokaryotic cells, certain ones have cell walls. There are bacteria that have a cell wall, and there are plants that have cell walls. But animal cells, which are eukaryotes, do not have a cell wall. So only certain prokaryotes and only certain eukaryotes have a cell wall. But because you find it in both pro and eucamp, we put it on the very mad CC list. That brings us to the endosymbiotic theory. The endosymbiotic theory. A long, long time ago, in a galaxy very near you, there was a very large prokaryote swimming around. And in this universe, it's a prokaryote eat prokaryote world. Big prokaryotes eat little prokaryotes. It's the way things are done. And this particular big prokaryote, about 1 billion years ago, decided to eat a little prokaryote. Now, when big prokaryotes eat little prokaryotes, they usually digest them, but this one didn't. It ingested but did not digest this prokaryote. Instead, the little prokaryote was allowed to exist inside of it. Over time, this little prokaryote, being protected from other big prokaryotes and getting everything that it needed in terms of energy and materials from the big prokaryote, this little prokaryote became highly specialized because it didn't have anything else to do. And in one case, it became highly specialized as an energy maker. It made ATP. In fact, it made ATP so well that it made this big prokaryote, it gave it an evolutionary advantage. And another little prokaryote became really good at making glucose. And over time, it gave that big prokaryote a big advantage because it always had energy. And over 1 billion years, the little prokaryote became more and more specialized, but also more and more dependent on the big one, to the point where it became a membrane-bound structure with highly specific function. You see, endosymbiotic theory explains how we developed organelles. Endosymbiotic theory explains how prokaryotes evolved into eukaryotes. And to finish up this lecture, the last thing are simply the levels of organization. Going from small to big, we start with cells, the smallest living thing. If you have a group of cells doing the same thing, working together, they're called a tissue, if it's the same type of cell. If you have a group of tissues all working together for the same function, it's called an organ. If you have a group of organs all working together for a living thing, it's called an organ system, and that living thing is an organism. So basically, these levels of organization show increasing level of structure, starting from smallest to largest, going this direction until you reach the highest level of structure, which is an organism. That's our BioPrinciples diagram, and I'm going to end the recording here. I'll see you guys on Thursday at 10 o'clock. Thank you much and have a great week.